Good morning, Accelerated Biology students. Mr. Adair here, starting off week 3 of quarter 3. We're going to start with a new topic today, and it's going to piggyback on what we've been learning about with our DNA molecules. So last week we learned about DNA, and now this week the structure of DNA, and you learned about Watson and Crick and Rosalind Franklin. And A's go with T's, G's with C's, nucleotides, all that good stuff. Now, this week we're going to talk about how DNA does DNA replication. And replication is just a fancy word for making a copy of itself. So DNA needs to make a copy of itself for one of the main reasons, if you look closely at this image here, we have learned that, our cells need to divide, and our cells make copies. Your cells are dividing right now. They go through the G1 phase, the S phase, the G2 phase, and then they go through mitosis, PMAT, prophase, metaphase, anaphase, telophase, and then finally cytokinesis, where two new cells are formed. But in this process, the DNA makes a copy of itself, and we call that DNA replication. So DNA needs to make a copy of itself for when cells divide, basically. And it needs to do this very perfectly, otherwise we can have major problems in the body. So a few basic definitions for you. You can read through this, but the process by which DNA is copied during the cell cycle, we call that replication. It makes sure that every cell has a complete set of the identical genetic information, the DNA. And you have in every single one of your cells a complete set of the DNA that you received from your parents. I should say in every one of your somatic cells or body cells. In your gametes, the egg and sperm, that's a different story a little bit. All right, so how does this work? Now there's a lot of words on this slide. Don't get too overwhelmed. But look at this image. It shows a DNA double helix kind of cartoon image, but it is unwound in that middle section, and we see the C, G's, and A, T's. So basically, what you need to, and then look at the bottom part of this image, the DNA further unwinds, and the original DNA is used as a template for making a new strand. We're going to talk about some of the enzymes that help make this process possible. But what we have here is that the DNA will unwind and then start making new strands of the DNA. It's as simple as that. We use the original DNA to make a new DNA. So the first bullet point says DNA stores information. And we're going to talk about this more, but when we say DNA stores information, DNA is a code. Just like a computer code is ones and zeros, the DNA code is the base pairs C, G, A, T, G, C. Now, all those, the order of the base pairs. So if you look at this on number 1 of this image, we see that on the left we have C with G, then we have A with T, G, C, T, A, G, C, so on and so forth. The order, or in other words, the sequence of that string of letters determines what that DNA codes for, what proteins it makes. And we're going to learn more about that next week. But essentially what I'm saying is these letters, the adenine, guanine, cytosine, and thymine, are the code for making life. It's pretty miraculous when you think about it. But and so all those physical features that you have, your eye color, hair color, height, everything is determined by the order of these letters in your DNA. So my order of letters is unique to me, and your order, when I say order, I mean sequence, same thing, is unique to you. Okay, so for this process to start, for when a cell divides, let's say right now my skin cell is going through cell division, it needs to make a copy of a DNA for the new cell to have an exact copy. What happens is that we have these enzymes that unzip our DNA double helix. The proteins hold the strands apart. That's what you can kind of see in that bottom image. There's nucleotides floating around in the cytoplasm, or in the nucleus, I should say, and they're going to pair up with the DNA strands. And when the process is done, there are 2 new strands of the DNA. We'll look at some more images here. So, and there's going to be some videos you can watch at the end of this presentation to learn a little bit more. The DNA molecule unzips as nucleotide base pairs separate. Helicase is the enzyme that unzips the DNA molecule. So I always remember helicase as the enzyme that unzips, kind of like you unzip a suitcase. In my brain, helicase kind of sounds like suitcase, and I know that you need to unzip a suitcase. So just another little trick to help you learn that. And replication begins on both strands of the molecules at the same time, and goes in both directions too. We'll talk about that. So there's a lot of these images, and they're all kind of showing the same thing. The main point I want you to get is that, especially look at either side of these. We have the DNA that unzips, and then, especially if you look at the picture on your right, we have our original parent DNA strand towards the top. Then you can see it unzip, and we have in the red and pink color the new strands forming by using the original template. Okay? And you're going to practice doing this as well. And you please watch the videos at the end of this presentation that will go through it in more detail. All right, this image is really good because it adds a little bit more information here. And again, we're going to go through this again. The 2 enzymes you need to know are, for one, helicase, which you see right at that replication fork. So if you need to pause this video and take time to look at it, you should. The replication fork is where the original strand of the DNA will be separated by helicase. Remember, you unzip. Then we have a molecule called DNA polymerase that adds the new nucleotides to the strands that have been unzipped. And A always goes with T, and G always goes with C. In this case, we have one strand, the lagging strand, that goes in one direction, and then the leading strand that goes in a different direction. This allows the DNA to replicate itself in both directions at a very fast rate. So this picture, take a look at it. If you look at the original parent strand, this is kind of what I was saying. We call DNA replication semi-conservative because you're conserving half of the original strand. And in this picture, the red is the new strand and the purple is the original parent strand. And the last thing, before I get into it a little bit more, is that DNA is going to go in both directions in multiple places on the long, super long DNA molecule. It's hard to conceptualize and think about, but DNA is like billions and billions of base pairs long. So in order to replicate the whole molecule, it's a big deal, and it goes through the process at many different places. Okay, all right. So these are the videos that you need to watch. I will post not only this video tutorial on our Schoology page, but I'll also post the original presentation, a link for it to the Google slide, where you can access the videos that are very helpful. All right, I'm gonna get out of here. I'm gonna open up my Jamboard real quick here and kind of go through some of this in a little bit more detail. So here's Jamboard. Let's see, I want to edit this just so I can draw on it a little bit. Here's our first image. Remember, now the DNA, the helicase separates this. The C of this strand is going to make a new copy of DNA using this information. So C goes with G, A, T, G, C. And this strand will also do the same thing, making this information. Okay? The strands will be identical when it's finished. So just a couple different pictures to look at it different ways. Here's our parent DNA strand. It unzips, and then in both directions we have DNA making copies. All right? And we're going to practice this. This was what I was talking about. The key vocab words on this are helicase enzyme. We have our DNA polymerase. We have our base pairs, and we have our leading strand going in one direction and our lagging strand going in the other direction. So make sure you spend time looking at this, and remember that what we're talking about is DNA molecule, but when we wind up our DNA molecule tightly with proteins, that's what forms our chromosomes. So when we say you got, your 46 chromosomes from your parents, we're really talking about your DNA. Another image of this, we see that two identical strands of DNA. We call this semi-conservative because we use the original DNA strand. Half of the original DNA strand in purple, that'd be the original, and then the red ones would be the copies. So we have two strands fully from one. Okay, your assignment is going to have some review questions, but it's also going to kind of look like this. So when you open up your assignment, you're just going to practice making a new strand. And don't overthink this. This says you're going to take the original strand of one and the original. This is like the process of dividing this out. So in original DNA strand 1, we have A. I got that from over here. C from over here. T, T, A, C, G, C. And I'll let you do this on your own. Obviously, you need to do this on your own. Then the original from, this is like when it divides out, the helicase unzips it and they separate. TGA. Now I'm getting these from over here at this point. TGAA, TGC. And now these are our original, and that's why we say this is semi-conservative, because it keeps half the DNA. Now the new strand is formed. Let me just use a different color. And we still use our base pairing rules. A, T, C, G, T, A, T, G, C, G. I'll let you guys do the rest on it. For below, C, G, C. Now, if you look at, these are our two new strands, right? Our two new strands of DNA. Boom, boom. Are they the same as the original? A, T, C, G, T, A. Yes, they are. So we just did a crash course in DNA replication. So watch this video, review the notes, and we'll go from there. All right, everyone have a great day.